Hello, ladies and gentlemen. It's my great pleasure to talk about our research. I'm Irena Rektorova, professor of neurology and head of the Movement Disorder Center, but also head of applied neuroscience research group at SATEC in Masaryk University. And this is the research which was done by my colleagues in the group, and it will deal with effects of dense and non-invasive brain stimulation on brain function. So I will now briefly touch upon the diseases because we enrolled subjects with both Parkinson's disease and Alzheimer's disease. So what is Parkinson's disease? It's a motor disease, although some non-motor symptoms as well as cognitive problems occur later on during the disease progression. So it is diagnosed with Parkinsonism, which includes bradykinesia, plus at least one symptom, which is muscle stiffness, we call it rigidity, a resting tremor, and postural instability with falls and gait problems. Now let me briefly touch upon an examination which is called DET scan, and it's done at the Department of Nuclear Medicine, and it shows degeneration of substantia nigra, which is subcortical structure, and you can see decrease of dopaminergic cells. On the left side, it's a normal picture in a healthy volunteer, and on the right side, you can see decrease in a patient or decrease of dopaminergic cells based on this examination in a patient with Parkinson's disease. Now, let me change gears to Alzheimer's disease. It is impairment of recent memory, orientation in space, but other cognitive functions are also involved. Later on, dementia develops, changes in behavior, and loss of dependency of the subjects. We can find brain degeneration, which means loss of neurons, especially in hippocampus, which is a memory-related area, but also in other brain regions. We also see changes in neurotransmitters, which is important for declarative memory and also for treatment, but up until now it is untreatable disease. Now let me show you the brain changes in Alzheimer's disease, and you can see senile plaques, which include beta amyloid, and neurofibrillary tangles that include hyperphosphorylated tau protein. I will also briefly mention cognitive reserve, which will be discussed later on. There is a huge variability between cognitive performance in healthy and pathological aging, and there are so-called superagers who really show increased thickness of anterior cingulate and increased density of so-called von Economo neurons. And these superagers cognitively perform as well as their 20 years younger peers, despite the fact that they can have already brain pathology of Alzheimer's disease. My colleagues will also mention non-invasive brain stimulation techniques, so I will not go into details in this, but you will hear about it later on. And let me touch upon aerobic training in seniors. It's well known that there is an adult hippocampal neurogenesis, especially in specific parts of the hippocampus, which is happening in neurologically healthy subjects throughout the whole lifespan. But it drops sharply in patients with Alzheimer's disease. It has been shown that physical exercise, especially aerobic training, works towards neurogenesis and enhancing neurogenesis in human adults and even those with brain pathology. This can be done with angiogenesis, symptogenesis, increasing of specific brain factors and growth factors. So it has been shown that physical exercise promotes recovery of synaptic plasticity and cognitive functions such as learning or object recognition. We performed dance movement intervention in healthy seniors and patients with mild cognitive impairment, and 100 subjects were randomized into two groups. One of them were practicing dance movement intervention 3 times per week for 1 hour at the Department of Sports Studies for half of the year, and the second subgroup of subjects performed life as usual without this intensive training. And we found a lot of results. One of them was increase of cortical thickness in areas that are associated with motor learning. Also increased structural connectivity, an enhancement of specific tracts, which are again important for motor learning. We also found changes in resting state functional connectivity within specific brain networks, various cognitive networks, motor network, visual network, and so on, and especially increased inter-network connectivity between these specific networks were related to improvement in attention, executive functions, and concentration that we showed due to dance as compared to the group which had life as usual. So now you will hear presentations of my colleagues from Applied Neuroscience Research Group from SATEC Maastricht University that will follow, and it will be Dr. Luboš Brabenec who will speak about non-invasive brain stimulation for hypokinetic dysarthria in Parkinson's disease, then Monika Pupíková and Patrik Šimko who will touch upon transcranial electrical stimulation for modulation of cognition, and lastly Kristina Miterová who will inform you about cognitive reserve and dance intervention induced brain changes. Thank you for your attention and goodbye.
Ladies and gentlemen, I want to tell you something about our research that was focused on long-term effects of transcranial magnetic stimulation on speech of patients with Parkinson's disease. Hypokinetic dysarthria is a quite common symptom in Parkinson's disease. Patients with Parkinson's disease usually speak very slowly, their speech is very quiet, and they also have some problems with prosody and articulation. Actually, there is no treatment for these speech problems. Therefore, we want to try some new approach on non-invasive brain stimulation. In our studies, we usually used repetitive transcranial magnetic stimulation. Repetitive transcranial magnetic stimulation is a non-invasive method that modulates cortical excitability, and it is based on principle of electromagnetic induction. In practice, a special electromagnetic coil is placed over the patient's head, and this coil sends rapid magnetic pulses, and these pulses activate and neurons below. Very important is placement of this electromagnetic coil. In our research, we use stimulation of supratemporal gyres because this cortical region is involved in the auditory speech feedback, and also previous research found that this region displays abnormal connections with other subcortical and cortical motor speech areas in patients with Parkinson's disease. In our exploratory study, we found that a single session of RTMS had an short-term effects on speech of patients with Parkinson's disease. In our longitudinal study, we focused on long-term effects of repeated 10 sessions of RTMS over the right superior temporal gyrus. We had two groups, one group with active stimulation and other group with sham placebo stimulation. All of our volunteers underwent an assessment in magnetic resonance and also an assessment of dysarthria by a speech pathologist. These behavioral and MRI assessment were performed at baseline before these 10 sessions with stimulation, and then at week 2, 6 and 10 after the baseline assessment. We found that these repeated sessions had a long-term effect on speech of patients with Parkinson's disease, and this positive effect lasted about 10 weeks after the end of the stimulation. And we also found that RTMS increased activation in remote areas of the dorsal language stream and also modulated functional connectivity of these regions. And also RTMS improved white matter integrity of anterior part of the arcuate fasciculus that is important in speech production. In our future study, we want to focus on combination of speech therapy and home-based stimulation using transcranial direct current stimulation. My colleagues Monika Pupíková and Patrik Šimko will talk about this kind of electric stimulation later. Thank you for your attention.
Ladies and gentlemen, let me introduce our next research, this time focused on cognitive modulation by brain stimulation. For this research, we used transcranial electric stimulation, which is a little bit different from the magnetic stimulation, and you can see it on a picture. So basically, the person has 2 or more electrodes attached to their head, and there is a current flowing between those electrodes. Usually, the person does not perceive almost anything. Just on the beginning of the stimulation, there are some itching or tingling that disappears after first 30 seconds of stimulation. As you can barely feel the stimulation, it's not painful. You can also perform some tasks during the stimulation, as you can see on the picture. There are 2 basic types of electric stimulation used in research and also in our research. The more basic one is direct current stimulation, and the other one is alternating current stimulation. If we simplify it, we can say that under the anode of this stimulation setup, there is excitation of neuronal firing under the anode, and under the cathode there is inhibition of neuronal firing and therefore brain function. in comparison to magnetic stimulation, which really fires action potentials in neurons, therefore activates neurons directly. Electric stimulation only enhances or changes the probability of such neuronal activation. The effect of single session of TDCS or TACS lasts approximately 60 minutes, but it can be enhanced by multiple sessions of stimulation, and the effect onset is after first 3 minutes, so it takes some time before we see stimulation effects. And now I will show you some research where we use this kind of stimulation in our studies. In the first pilot study we used TDCS stimulation in young healthy volunteers in age of 27 years. We tried to modulate visual attentional task, which you can see on a picture below, which consisted of pairs of objects, and our participants were supposed to say whether the objects on a picture are the same or they're different. On the first rows you can see a simpler version of the task from the same point of where objects are in the same point of view. On the lower two rows you can see a little harder version of the task where the objects are rotated, but the task is the same to say whether the objects on the picture are the same. We use this task before and after each stimulation together with fMRI. Each participant underwent real and placebo stimulation on different days, and the order of real and placebo stimulation was randomized. During the stimulation, we used another visual attentional task that you can see on the picture with scenes and faces. Subjects were supposed to remember blocks of 4 pictures, and after a short retention period, they answered whether the face or scene was in the previous block. We used this task in order to activate brain areas or brain networks that are involved in such a task, and to make the stimulation more effective, more specific for such a task. For brain stimulation, it's very important where you place your electrodes. In this research, we used frontoparietal electrode montage, which you can see on a picture. Anode was over the frontal lobe and cathode was over the parietal lobe. These brain areas are hubs or parts of very important larger frontoparietal control network that is involved in visual attentional tasks. For being precise in placements of electrodes, we use stereotactic navigation together with MRI scans of each individuals, and that allowed us to be very precise in electrode placements. And now we get to results of our study. We showed, or we saw that reaction times of our participants in the visual object matching tasks were much quicker, significantly quicker after real stimulation compared to sham or placebo stimulation in the harder version of the task, that means in the rotated view of objects. For accuracy, the accuracy was very high already, and there was the roof effect, so there couldn't be found any effects. That means that even in young healthy volunteers whose brain functions are optimal already, we can still modulate brain functions by TDCS.
The objective of the next part of our study was to test the most effective protocol enhancing visual attention in older adults. We enrolled 27 healthy seniors aged around 17 years. All of these subjects underwent both real and sham stimulation in a crossover design. The study was double-blind, randomized controlled trial. We used the same attentional visual task as in healthy younger adults that was already described by my colleague Monika Pupikova. We focused on the after effects, so the short-term after effects immediately after the end of the stimulation. We compared two protocols, one the frontoparietal protocol, enhancing the right hemisphere, or stimulating the right hemisphere, and the left bilateral or bifrontal protocol, where the anode was placed around the left dorsolateral prefrontal cortex, which is the one of the most used nodes for enhancing cognition in other conditions, such as for enhancing executive function and so on. What we found was an increased or enhanced accuracy after the bifrontal stimulation, but not after the right frontoparietal stimulation. As you can see on the right bottom corner, there is a graph which shows that after the anodal stimulation, there was a gradual increase in the accuracy, and compared to the blue line, you can see the placebo stimulation, there was almost no change at all. So we can say that the visual attention was enhanced in older adults, despite the fact that there is a gradual reduction and deterioration of brain plasticity and cognitive functions in older adults or coming with older age. Moreover, we were interested in the neural underpinnings of the elicited brain plasticity changes after the TDCS stimulation. We used functional fMRI before and after the stimulation to see the changes after both the real and the placebo condition. What we were able to see was an increased intra-network connectivity within the frontoparietal control network. That's a large-scale network that's function is to control cognitive functions. An enhancement of the connectivity within the frontoparietal control networks shows support for enhancement of cognition. There is also a theory that people by getting older have more less distinct processing within the large-scale network. So enhancement of the within network connectivity is a sign of youthful pattern of brain connectivity that was induced by the stimulation. In our next part of our project, we will be interested in applying the same protocol on the MCI patients. So those are patients who already have an objective problem with memory, and most of the time the pathology behind the memory problems is Alzheimer's disease. We will be interested in the long-term effects by applying multiple session stimulation during 1 week, at least in 5 sessions. Furthermore, we are interested in the transfer of these effects, so whether the elicited effects are more generalizable to other cognitive domains such as episodic memory and navigation, spatial navigation. We will use for this virtual reality task where the subjects have to navigate through the supermarket and remember a specific shopping list. In other projects, we are interested in enhancing working memory, which is a short-term storage or buffer for different objects. According to the physiological background, the working memory is governed by theta, slow wave theta activity, and fast wave gamma activity. According to the theta gamma nesting theory, the more gamma fast waves can be nested within the one theta wave, the higher is the working memory capacity, as shown before by Waxel et al. In 2015. Taking into account this, we want to use alternating current stimulation to influence the working memory. With this aim, we want to use a multifocal type of a stimulation by stimulating multiple nodes within the frontoparietal network, and thus more respecting the physiological background of the working memory processing. Furthermore, we are interested in individualizing the stimulation. We want to achieve this by spatially individualize the stimulation by scanning first people and identify activation maps underlying working memory processing, and then use these maps as targets for the electrode placement. Furthermore, using electroencephalography, we are interested in the endogenous oscillatory activity underlying working memory, and thus we can individualize the frequency for the stimulation. In a stepwise manner, we want to combine all of these principles together to even more customize and phenotypize the stimulation to have higher effect sizes. From design perspective, the study looks for following. You can see on the upper picture, right before and right after the stimulation, the people are being scanned in the fMRI machine. And so we are interested in the changes on the brain activity and brain connectivity. And furthermore, we want to understand whether the effects are more generalizable by virtual reality task. Thank you.
Dear ladies and gentlemen, in the next couple of slides I would like to present to you about the topic of cognitive reserve and its effect on dense movement intervention outcomes. So first, let's define cognitive reserve. It can be divided into neural reserve and neural compensation. The first part is associated with capacity and efficiency of a healthy brain. So for example, it is also correlated with intellectual functions or cognitive functions in a healthy brain. The second part, neural compensation, is the ability to actively respond to a damage or a pathology. So for example, on this picture below, you can see that the person B, despite having substantial pathology, which could be visible by looking at larger ventricles or loss of gray matter, despite that, they reach the same cognitive score as the person A. This could be ascribed to their higher education. Education, together with occupational complexity, leisure activities, regular exercise, intelligence, or premorbid brain volume are one of the most important determinants of cognitive reserve. In other words, persons with a higher education, for example, are better able to withstand more brain pathology and have a reduced cognitive decline. So in the next couple of slides, I would like to show you our two research which were focused on potential moderating effects of cognitive reserve on a dense intervention outcomes. So what do I mean by moderation effects? We were hypothesizing that people with higher cognitive reserve or higher education would benefit maybe more or maybe differently from these kind of interventions. Dense intervention in and of itself has been shown to improve memory or other cognitive and executive functions in seniors with mild cognitive impairment or just healthy seniors. We were focused on the following outcomes. Firstly, behavioral changes, so cognitive functions and physiological fitness. Secondly, we studied changes in brain functional connectivity. In the first study, we were specifically interested to see changes in sensory motor network and between default mode and dorsal attention networks. The second study studied overall changes in brain functional connectivity. We proxied cognitive reserve by education in the first study, and in the second study with residual index, which is more sensitive to changes due to pathology or cognition. In the first study, we specifically observed increased connectivity in seniors with higher education. This connectivity was improved in sensory motor network and between already mentioned default mode and attention networks. These changes in brain functional connectivity were also supported by improvements in cognitive tests and motor fitness tests. On the other hand, results of our second study showed that also elderly with lower residual cognitive reserve are able to benefit from such dense intervention by increasing time in a bottom-up brain state, which supports salience perception. In other words, these people improved in their psychomotor speed. Elderly with higher residual cognitive reserve, on the other hand, again improved in processes of top-down cognitive functioning, which is associated with prior knowledge. In conclusion, our two studies showed that seniors benefited differentially from such dense intervention. The benefits really depended on their current status of cognitive reserve. So people with higher cognitive reserve were able to improve their higher cognitive functions, such as executive functioning and motor functioning, whereas persons with lower cognitive reserve benefited more in terms of increasing or improving their psychomotor speed. In the end, I would like to thank you for your attention, and I hope that we inspired you not only to improve your cognitive reserve, but maybe to also follow science and participate in research. Thank you.